Welcome everybody to our webinar series, Critical Transitions in Complex System, what we call CTCS. Seminar series is being jointly hosted by IIT Madras in India and Potsdam Institute for Climate Impact Research, that is PIC, in Germany. This seminar series brings together experts from diverse fields such as climate science, fluid mechanics, neuroscience, ecology, etc., and aims to disseminate the state of the art in the prediction of critical transitions in these diverse fields of study. So seminar usually takes place every month on the last Monday. Today is an exception because of some scheduling difficulties. So please remember that this is a monthly event. And before we introduce the speaker, a few housekeeping notes. One, I request everyone to turn off their microphones so that the talk goes smoothly. And I request the audience to use the Q&A box in Zoom to ask the speaker your questions. And we'll have enough time at the end to discuss all the questions and clarifications. With this, I invite Professor Jürgen Kurz to introduce the speaker.
Yeah, thank you very much, Sujit. I'm really very happy to introduce to you today Professor Plamen Ivanov from Boston University, from Massachusetts, US. We have known each other for a long time. I have not counted the number of years, are very good friends, have a lot of joint scientific interests. And Plamen started with basic contributions on really fundamental aspects of statistical physics, multifractal description, and several others. And then he realized that is not enough. He also would like to go deeper to applications. And he has not so recently introduced a new discipline and really worked out this. And this is called network physiology. This is really, you see, is really interdisciplinary, combining now network science, statistical physics with problems in physiology of highest interest. And this is really a very modern aspect. And he has also a journal on this and has more and more people interested and working on this. And I'm really very happy that he will introduce the basics on this, but I think also modern achievements. Plamen, you have the floor. Thank you very much for being able and being willing to give the talk.
Thank you very much, both Jürgen and Sujit. Really, it's a pleasure for me to be invited to be able to present at such a distinguished webinar series, to speak in front of audience in India and also in Europe, and to have the opportunity to present a work related to indeed a new interdisciplinary field called network physiology, and how this could bring better to understanding fundamental questions in medicine and in human physiology, how we can develop the human physiolome. I'll explain a bit about that by implying concepts and approaches from computational and statistical physics and nonlinear dynamics. So thank you for this opportunity. I really appreciate it to be able to speak here. So we know that the human organism comprises various organ systems. We have the brain, the heart, the lungs, kidneys, muscles, eyes. All of those systems are very, very complex systems. In the quintessential definition of a complex system, they have a complex structure that is still being investigated, and of course, a very complex dynamics and again, complex mechanisms of physiological regulation behind, involving multiple scales, multiple feedbacks, and their focus of investigation over many decades. And still we have a lot to learn about these systems, their structure, their dynamics and function. And even more interestingly, it's not just that they're simple structure and simple dynamics, but it's a lot of network dynamics is involved in the regulation and the function of these systems. There is an example of the heart. This is the vasculature network of the heart. And surprisingly, there is no mathematical model or understanding of the branching patterns here and how this facilitates the heart to work. Of course, on top of this vasculature network, there is also a network of myocardial cells, very anisotropic, very complex. This is the heart muscle. And inside the heart muscle, there is another network. This green type of patterns that you see, this is the network of the Purkinje fibers. These fibers are absolutely essential to conduct electric impulses through the heart so that the myocardial cells can contract and the heart can pump blood. So we still have no understanding from network and dynamics point of view of the structure and function of this type of vasculature network or the Purkinje fiber network. Another example, take the kidneys at different scales of observation. At a larger scale, smaller scale, you start to see the cortex of the kidneys. And here at a smaller scale, all the branching patterns of the various structures up to the level of these small blobs called glomeruli. And so there are millions of glomeruli in each kidney, and they somehow work as a network in a synchronized way. There is still neither empirical studies or models to understand how the kidneys manage to synchronize all these different elements together so that we efficiently purify blood. And by the way, the kidneys use these networks to purify blood, bigger, larger volume even than the volume that is pumped by the heart. So if you go to the lungs, it's another network. And actually the lungs have two networks also superposed. The airways network, here shown to the left, and also the vasculature network. So there is a spatial organization of these networks. A lot of work in the last decades has been studied both structurally and dynamically in this network, but they branch down up to the alveolus. And the alveolus themselves are a very intricate network, almost no studies at this level. Of course, if you go to the brain, especially in the last decades, a lot of function is there, but still we have, this is the imaging, tensor imaging of the brain with all the fluidity information and different bundles of and structural dynamical networks. Here, at the same time, it's also superposed with the vasculature network. And indeed, it's also superposed with the subnetworks of various, neuronal cells with different topology and different function. And this is a real network of about 300 neurons and electron imaging of their activity in real time from a Harvard Medical School lab. So this is a good justification to say that having the task to understand the structure and function of individual systems is an immense objective and requires a lot of effort, and not by surprise in the last hundreds of years in scientific research, both in basic sciences and in physiology, people have focused on individual systems and trying to understand their individual complexity, individual structure, and mechanisms of regulation behind. So neurologists focus on the brain, and neuroscientists, cardiologists on the heart, pulmonologists on the lungs, and people, ophthalmologists on the eyes and so on. But we know that the human organism is actually an integrated network of all these systems and subsystems. And these systems continuously communicate, they interact at various feedback and various timescales in order to maintain health. These interactions are absolutely essential. If we break down these interactions, we cannot generate health and distinct physiological states. And indeed, if you impair, for example, the breathing, stop breathing for a while and you see your heart rate starts racing. So dysfunction of one system can lead to, through the network of interactions and various forms of coupling between systems, can lead to the dysfunction of other systems and ultimately to the collapse of the entire organism. So now failure of one system may trigger a cascade of failures in the entire organism, and there are many critical states and conditions of the human organism that exhibit exactly this, and they're subject of treatment in intensive care. On the other side, you may have individual systems fully intact and structurally intact, and still this is not sufficient for health. For example, coma or multiple organ failure very often people come to intensive care unit with this condition, multiple organ failure, first symptoms related to one system, then to another. And after autopsy, it turns out structurally systems are intact. Then the question is what triggered this failure? And apparently something in the way how these systems cross-communicate was interrupted. So despite the importance to understand basic physiology and the immense clinical relevance for health and disease, today we still do not know how organ systems dynamically interact as a network, how they really coordinate their individual functions, how they optimize their dynamics in order to generate states at the organism level. So each organ has a separate scale of action, separate type of distinct dynamics, yet they coordinate somehow together. At the level of the organism, we have behaviors that are not similar to any one of the parts. And so in the last 20 to 30 years, systems biology and integrative physiology, these disciplines have focused on the signaling and the feedbacks across space and time scales from the subcellular to the cellular and tissue level, and how different subcellular elements from genomic and proteomic components to cells and tissue, how cells cross-communicate in a tissue and integrate vertically up to the organs. But we still do not understand what is the horizontal integration among key organ systems, and how out of this vertical and horizontal integration distinct physiological states emerge. So a state of wake or a state of sleep, the state of rest or the state of exercise, the state of anxiety, the state of cognitive function, hundreds of various physiological states, state of fatigue, for example, related to exercise and so on, and how they emerge out of such complex vertical and horizontal integration among systems. And particularly here, this area of horizontal integration among organ systems and their sub-components is practically, we realized about 15 years ago, it's practically not investigated. And fundamental question is, would there be a particular network structure and particular network dynamics, a particular blueprint reference map that uniquely defines a physiological state? So in order to generate a given state at the organism level, do all these dynamical complex systems need to adjust in certain way to form a particular network with a particular dynamics and topology in order to facilitate the emergence of the state? And then, of course, how these networks change in order to trigger disease. And so these are really fundamental questions that made us realize that we are at the verge of a new discipline, because every discipline has a unique set of fundamental questions that require a new set of theoretical approaches and theoretical frameworks. So we, about 15 years ago, in 2008 and 2007, we embarked on this new research direction, shifting the focus of a single system, even though we recognize that we still do not fully understand the structure and dynamics and the mechanisms of regulation of individual system, but still we realize that we need to start to see these systems through as a network in the form of integrated behavior to understand how the organism function. And so we realized there is a need of new field called network, we call it network physiology, that needs to probe the interactions among diverse physiological systems very much in this Leonardo da Vincian way, where the human body is considered by, let's say, key nodes here, and the nodes represent key dynamical systems. And these interesting links, they are not static links like in the complex network science that was developed very often in the last decades to address various systems. But here we learned, and I'll show later, that these nodes are dynamic and the links of interaction are dynamic. So this really poses a new challenges to theoretically approach and also computationally dissect this data to understand better what is behind. So now there are many challenges, and here the audience obviously comes from a background in nonlinear dynamics, statistical physics, complexity, phase transitions, and of course biology. So we have levels of complexity at the individual system, because individual systems, as I mentioned, are complex structurally and dynamically, but specifically from dynamic point of view, their output signals are very noisy, non-stationary, and they change in time. There are transitions in the dynamics of individual systems, and I'll show a couple of slides, examples about it. In addition, there is another level of complexity, the interactions between physiological systems. They're pairwise coupling, and their pairwise coupling is also nonlinear, also transient, related to certain transitions when you go from one physiologic to another state to another. This pairwise coupling between systems also undergoes a transition. Actually, it's a trigger for the transition across physiological states. And on the top of that, there is a third level when we have multiple systems together, each one with their individual dynamics, with their individual scales of interaction and pairwise coupling, how together as a network they lead to certain global behavior at the organism level. And it's, of course, a multidisciplinary approach that requires new concepts and new theoretical framework that would integrate data-driven discoveries both in computational science and statistical physics and applied math, but also medicine and physiology. So now here are a couple of points related to the dynamic complexity at different levels. First, at the level of individual systems, and in the last 30 years, especially at the end of 80s, beginning of the 90s, people started to notice that individual systems, no matter the scale of observation, whether it's at the subcellular level, at the cellular or the organism level, they exhibit dynamics characterized by complex fluctuations. And if you take, for example, subcellular level, this is a single ion channel, a protein that transcends the membrane of a cell. This protein changes configuration very frequently so that the conduction current across the membrane changes in time. And so this is the time series of the ion channel kinetic, and one can see it's a very complex, fluctuates, and it looks at first glance very random. But if you take a single spike here and you change the scale of observation, you magnify it, you'll find that this complexity of fluctuations goes down and down to smaller scales. The same is true if one considers the fluctuations of individual neuron. And so we have this complex Morse code or pattern of bursting and quiet states of neuronal activity. And people are trying to understand the dynamics and the complexity of this variability in the bursting of individual neurons. And when we integrate millions of neurons as a network at the level of the brain, as a system, we can measure EEG signals, and these signals are very complex also. So we have fluctuations across scales, and people in the last decades have realized that these fluctuations are not just a perturbation to the systems, but they carry a very fundamental information about mechanisms of regulation behind these systems at various levels. So here is one example, comes from our lab way back 30 years ago. If you take a EKG recording and your heart rate beats, and when it beats, it performs these waveforms called QRST complex, and this is what usually clinicians look at, and when they see change in this waveform, they can say something is wrong in your heart. But one can see also the interactions between consecutive heartbeats. They are called these R peak to R peak interactions, and they are plotted here as a time series, and people have found that this time series is quintessential expression of complexity, because in addition to the variability, it's characterized by various trends and also by changes in the standard deviation, by changes in the amplitude of the variations. So the question is there any information here? And way back, this was a hypothesis 30 years ago, and it was found that if you dissect this noisy signal across various scales, large scales of observation on the vertical axis to smaller and smaller and smaller scales, and this is on the horizontal axis is the time, one can see that behind these fluctuations there is a very complex bifurcating cascade of bifurcations going down from a larger scale, bifurcating to another cascade, bifurcating to smaller and smaller. You take a small window here and you see it goes to a smaller scale. So there is certain self-similarity here and a certain temporal organization. And therefore, there is also a mechanism behind that regulates this so-called fractal or multifractal type of characteristics that break down with disease. So this is one example of how complex are the dynamics of these systems, and how this complexity is also characterized by important temporal structures across scales that may break down with disease, and then this breakdown can become, here the pattern, you can see the same pattern here is replication from a cardiac data from a subject with sleep apnea, and it's very, very periodic. There is no cascade, self-similar cascade, and so therefore you have a different altered mechanism of regulation and new type of diagnostic tools. So now when we go to the second level of pairwise coupling, one classical example is the coupling between the respiration and the heart. And as I said, if you stop breathing, your heart rate will stop racing. And way back about in the 1960s, Angeloni and Coulter have observed that when you breathe in, inspiration, your heart rate goes up. And when you breathe out, expiration, your heart rate goes down. And since then, there are hundreds of thousands of clinical studies exactly focusing on this phenomenon, how the heart rate variability and the heart rate goes up and down in synchrony with inspiration and expiration. Why? Because this pattern can also change with age, as it's shown here by these other curves. And the change in this pattern, therefore, is a indicator of certain physiological state or clinical condition. But the behavior is even not so trivial, because it is nonlinear. This dependence of synchrony between the breathing and the heart rate is a function, it's a very nonlinear function. This nonlinear curve is a function of the frequency of the breathing. They are aligned in this way, up and down with inspiration, expiration, only when you have a particular range of breathing. When the breathing declines, this ratio becomes very nonlinear. This is an example, actually, and here there is another curve showing. Here is exactly when they are aligned, but you can change the breathing pattern on the horizontal axis, and the synchronization can be exactly the opposite with 360 degrees, and then the heart rate will increase when you expire and decline when you inhale. So this is really a complex behavior. Just to show, it's called respiratory sinus arrhythmia, one form of coupling between two very complex dynamical systems. Now, on top of that, pairwise interaction between systems is characterized by multiple forms of coupling. This is one that I just demonstrated. It's called respiratory sinus arrhythmia, what most clinicians know. But about 20 years ago, colleagues, Professor Kurtz and Rosenblum and Pikowski, there is a famous book called Synchronization and Adjustment of Rhythms of Self-Sustained Oscillators that showed this type of interesting phenomena practically, that sometimes if you have independent oscillators but connect them even with a weak coupling, they can exhibit very interesting behavior by synchronizing their phases. And so this is what they demonstrate. You have metronomes which have same frequency but very different phase. So you see that they are working out of phase. And in time, gradually, they start to form pairs of moving in phase in synchrony, and then gradually, more time, you'll see that the entire set synchronize. And this is the locking phase. And so this is a very, very big discovery with big implications for understanding interactions of nonlinear systems. And even more, it turned out that it had big implications to understand coupling and interactions in physiological systems. And now here is the respiration in the heart. Here is a one oscillator, the breathing cycle, one breathing cycle and another breathing cycle and a third breathing cycle. And here is the second oscillator, the heart. And this is the one beat of the heart, the next and the next. And again, 3 beats and 3 beats. So we have 3 heartbeats per cycle. So it is a frequency synchronization. But on top of that, it turns out that the phase in which all these 3 heartbeats hit the first cycle are phase 1, 2, and 3. And the other 3 heartbeats also hit exactly the same phases, 1, 2, and 3. And the other 3 heartbeats hit exactly the same phases. So when you plot on the complex phase, so there is a synchronization. Always the first heartbeat hits phase 1, the second hits phase 2, and phase 3 for the third one. So one can plot these horizontal lines for this is the first cycle, the blue is the second cycle, the third cycle, but the phases align along these horizontal lines. So now this is quite surprising because if you look, this data is very complexly variable. This is the interbit intervals that I showed before. They fluctuate in time. The heartbeat intervals are plotted here, and the respiratory intervals are plotted above. And out of these very complex fluctuating signals, there is a transition of a time where we don't have phase synchronization, and then comes this segment of phase synchronization between the cardiac system and the respiratory system, despite their variability. So they look very much metronomic here when we look at this scale, but actually there is variability in the interval, there is of the heartbeats, variability in the breathing interval, and this variability adjusts in a miraculous way so that we have a synchronization that occurs on and off with certain transitions from period of no synchronization to periods of synchronization. And this turned out to have a very important physiological implications. So this nonlinear type of theoretical approaches developed by Pikoski, Rosenblum, and Kurtz turn out to be very useful to understand what is the physiological coupling between systems, for example, across different sleep stages. Here is REM, wake, light, and deep sleep, and one can see a dramatic increase, almost 400%, in the end-to-end phase synchronization between the cardiac and respiratory system. There is a very unique stratification pattern now that we learn underlies physiological pattern, underlies this coupling. And so we still do not understand the mechanisms behind, and this is a matter of investigation. But why is it also important? Because this stratification pattern, very stratified, you see here the very big difference between REM, wake, light sleep, and deep sleep. This pattern starts to decline in stratification when you go with increased age. So these are different groups of subjects in a younger age, middle age, and higher age of 80, above 80, about 50 subjects are represented here as a group average, and we see how this stratification pattern dynamically changes as a result of change in the cross communication between these two systems. Now, even more complex, I showed you that we have a respiratory sinus arrhythmia, this pattern going up and down when you breathe in the respiratory cycle, and we also have synchronization. Now, this is a data taken from the same subject in the same physiological state called deep sleep, and few minutes of deep sleep are observed when the subject shows the breathing cycle, and the black dots represent the heartbeats over multiple cycles. So these are 2 cycles shown from 0 to 4 pi, and but they're repeated multiple times over the several minutes, and the heartbeats are plotted. And the heartbeats are represented by these black dots. They are homogeneously distributed in the respiratory cycle. So we have the amplitude going up and down, and the entire cloud of heartbeats also goes up and down with the green line, which is the breathing. So we have respiratory sinus arrhythmia, but we don't have phase synchronization. And now here we have exactly the same respiratory sinus arrhythmia, the same green line going up and down, but now all the black dots representing the moments in which individual heartbeats hit the respiratory cycle, they're clustered in one phase, another phase, a third phase. Same phase, another phase, third phase. So there is both synchronization and respiratory sinus arrhythmia together. So now we have an example that physiological systems not only have individual complex dynamics embedded in their variability, not only that they interact with pairwise coupling that show transitions across physiological states, and this pairwise coupling can be characterized simultaneously by coexisting forms of interaction. But now we have a third level also of emergent global behavior, and that is not simply the sum of the individual behaviors of individual systems. And so, of course, change in the pairwise coupling in systems, in certain pairs of systems embedded in the network, can lead to a very different network dynamics, even when you don't change the network topology, the number of links or the number of the nodes. So currently, we realized some almost 2 decades ago that we do not have a theoretical framework to study this type of dynamic network interaction. So we approached the human body as this type of dynamic network of dynamic systems where different nodes, the heart rate, respiration, chin muscle tone, leg muscle tone, eye movement, different cortical rhythms that have various physiological functions, delta, theta, alpha, and sigma, and so on, can be, the question was, can we integrate all this together and see whether there is any association between the way how the systems interact and the corresponding physiological state that the organism is in. And so at that time, I was faculty at the sleep division at Harvard Medical School, Brigham and Women's Hospital. So sleep was a good candidate where we could record data simultaneously in a synchronous way, high frequency data over many hours, transcending various physiological states, and monitor the transition across states with transitions in the dynamics of individual systems. This means the dynamics of the individual nodes, as well as the transitions of connectivity between these nodes. And if you look as an experimentalist in this data and study at a detailed scale this data, one can see that this is a trace, a time series of the heart of the brain data, EEG, and particularly of the sigma rhythms embedded in the EEG. This is the heart rate interbeat variability. This is the respiratory cycles, interbreath variability. This is variability in the eye movement, just to show a few. And one can see that there is a synchronous in the bursting activity shown by this vertical line. When there is a burst in the brain, burst in the respiration, burst in the heart rate, in the respiration, in the eye movement. Again, burst in the brain, burst across all other systems. So we started to realize that there is a period of time in which these systems work in synchrony. So we decided to develop a concept called time delay stability, looking at the synchronous bursts in the time series of these signals, quantifying their cross correlations in small windows of time, and studying the maximum of the cross correlation at what time scale, at what time lag it occurs. So now these are the time delays of cross correlation between two seemingly synchronized signals. In this case, both signals come from the brain. This is sigma rhythm of the brain and delta rhythm of the brain. And so this is tau zero is the time delay between these bursting activities. So you can see that the time delay fluctuates randomly quite for a period of time, quite wildly, with a very broad range, and for some reason it stabilizes at some point. So the red dots here represent these 4 segments, where we can see also by eye that the bursts activity in these 2 time series synchronizes. So what triggers this synchronization? So now we realized actually that this was something new, because testing various other systems and other methodologies, how they would perform on output dynamics of systems with different bursting activity, but also different characteristics. So these are 2 signals with positive autocorrelations and synchronized bursts, and these are 2 signals with anticorrelations and also synchronized bursts. So when we compared methods, previously existing methods related to cross correlation, to transfer entropy, to coherence, one can see that all these methods would not respond to change in the burst density or change in the ratio of synchronized bursts compared to all total bursts in the data. So of course, if all bursts synchronize, then of course this ratio is 1, and we should have a very strong synchronization. And these methods will not pick this up in the presence of the noise, under background noise of these signals. But the time delay stability shows a very good sensitivity both to the density of such bursts and the ratio of synchronized bursts. So tested on Rukov generated oscillators. Here is 2 oscillators, neuronal activation called Rukov models. They are not synchronized. And here you see the bursts are synchronized. So you couple the models. Now, the time delay stability shows a very good response by increasing the strength of coupling, and also very good response by increasing the background noise. So there is one example of coupled oscillators without noise, and here is an example of coupled oscillators with noise. So this bursting activity can be picked up in the presence of very substantial amount of noise, up to 30 to 40% of noise compared to the amplitude of the bursts. And we find that the time delay stability is showing a very stable behavior and also sensitive. So this is all time delay stability measures for different degree of coupling in the Rukov system of coupling, of neuronal oscillator coupling. So this was a small deviation to justify for those who work in the nonlinear dynamics field that there is a way to develop, and there is a lot of space to develop new methodology that would be useful to identify coupling between systems that characterized by transient behavior, characterized by noise, and also bursting activity. So now coming back to the human body with multiple systems of interaction, let's take one pair, heart rate and eye movement. Why the heart rate should be synchronized with eye movement? There is no a priori reason for that. But let's take this random pair, and we can see the time delay, how it randomly changes for a period of time. So they are not synchronized. Their bursting activities are not synchronized. And at some point, something happens and they synchronize. They show a period of stable time delays. Take another pair, alpha rhythm in the brain and chin muscle activity. Again, why there should be a synchronization between these two systems is another question, but they show also a random bursting activity for a period of time, and at the same moment, they synchronize. And then we realize that this moment of transition is the transition between deep sleep and light sleep, dark area and the light shaded area. So if you take just these 4 minutes of small time period and take 30 seconds of time windows, how this network of interactions of system changes, one can see the topology gradually is one type of topology where we have very weak interaction between the brain and the peripheral systems. And now miraculously within few seconds, these are time windows from one network to another of 30 seconds, but literally within few seconds, there is more links appearing in the topology of the interaction and more systems become involved. And you see here the yellow and the red link corresponding to this stable behavior, exactly the transition from deep sleep to light sleep. And one can say, well, this is just by chance happening in this particular transition. Then take 6 hours of recording of the same subject. This in this case is 4 hours of data. And here the red curve shows the different sleep stages. On the bottom, the deep sleep, higher up the light sleep period, a bit higher up the REM, and on the top the wake. So always when we have a segment of deep sleep here, we have the black curve showing the connectivity of the network. Network connectivity shown with the black curve. Low connectivity in the segment of deep sleep, low connectivity segment of deep sleep, low connectivity for another segment of deep sleep, and low connectivity for another segment of deep sleep. Now we go to light sleep, and the connectivity increases dramatically. Light sleep and the connectivity is high up. Again, light sleep and the connectivity is high up. And we see this consistently across various states. Now this starts to indicate the presence of a certain basic physiological mechanism that was not known to physiology and medicine before in the context of sleep. So this work published in Nature almost 15 years ago and showed that different physiological states, basic states like wake, REM, light, and deep sleep, which are studied now for more than 150 years, are characterized by specific topology of the network interactions among organ systems. This topology has a stratification of the number of links. So high number of links for wake and light sleep, low for REM and deep sleep. So there are 2 columns here. Red bar shows individual subject, black bar shows a group average of about 40 young subjects, and we have a very interesting stratification pattern that was not known to science and to medicine before. In fact, quite counterintuitive, because if you ask every sleep specialist in sleep medicine, they will tell you that light sleep and deep sleep are very similar states. And we consider them similar, and REM and wake are also very similar. And this is true when we consider the dynamics of individual systems. But it turned out, from point of view of network interactions among systems, among organ systems, there is a completely different law regulating these phase transitions among physiological states, so that there is a very high connectivity in light sleep and low in deep sleep. Very different states from point of view of network interactions, and not only of the point of view of connectivity, but also point of view of the link strength, because we may change the connectivity, but how the strength of the link changes. Here, all links are presented in blue color, but their strength actually changes, and this is shown with a similar stratification. So both the connectivity of the network and the link strength, the strength of interactions between systems, undergo certain phase transition with transition across basic physiological states. Now one can ask a question, well, this may be idiosyncratic, some random reconfiguration of the links and the nodes interaction that goes together with transitions across sleep stages. But it turns out it is not random. If you take each node separately, it is the node of the heart rate, high connectivity during wake, lower for REM, high during light sleep, and almost weakly connected, they're almost disconnected during deep sleep, with the same pattern of average link strength, up, down, up, down. If you take another node, the chin muscle tone, how is it synchronized with the rest of the systems, the brain, the heart, with the leg, and so on? Again, high connectivity, low, high, low. At the level of each individual node of such a relatively small network, we have a very complex hierarchical reorganization with transition across states that is also paralleled by reorganization in the strength of the interaction. And no matter the node, whether it's the heart rate or the chin, we have the same pattern. So now we are aware that we actually discover a new physiological law. If you integrate this type of interactions across the brain, this is a graph representing two areas of the brain in the frontal. They called frontal areas, two central areas, and two occipital areas. Now, this is just a look only in the brain. Of course, people have studied various type of functional and structural anatomical brain networks, but what has not been realized is that cortical rhythms located, and these are different cortical rhythms, again, delta, this rosette represents different rhythms, delta, theta, alpha, gamma, at different locations, 6 locations, this can be also 60, just for the example here, how these rhythms cross-communicate with each other, how do they synchronize across various areas of the brain. So this is the left hemisphere and the right hemisphere, and one can find a very complex hierarchical network with various modules, one module here, another horizontal module here, stronger links represented with red and dark blue colors, and this network undergoes a transition across sleep stages, again with lower connectivity and REM sleep and lower during deep sleep, and higher for light sleep and wake. The same law as we have seen before. And this law is characterized by the connectivity, number of links up and down, cross states, and also the average link strength. Not known before, first not known that cortical rhythms communicate and that they communicate as a network following such a unique universal stratification pattern. These are data for a group average of about 50 healthy subjects in this young group of 25 to 35-year-old. So one can take and observe the same pattern in the subnetworks of the brain network, only looking at the left hemisphere, and one can see that deep sleep is associated with one single module, frontal to central. Light sleep, you add another module, central to occipital area. Then for REM, light sleep, you add a third module, frontal to occipital. And then for wake, practically everything gets connected. So the different modules are represented by these different bars, and we see that there is a very particular order of the bars, always stronger connectivity frontal to central, which is the first, bar, weaker connectivity central to occipital, which is the second bar, and of course the weakest connectivity is frontal to occipital. Still, for each module of this subnetwork, left hemisphere subnetwork, we have stratification. If you take the very first module, up, down, up, down. If you take the very last module, again, up, down, up, down. So this behavior is observed across various, globally in the entire network, but also across subnetworks and modules of the subnetwork, very symmetric across hemispheres also. So this can be observed in cross-hemispheric interactions, diagonal interactions, and so on. Now, if we extend this and we go to different organ interactions, how the brain, for example, interacts with chin muscle activation, this can be represented by this radar chart network. Again, 6 areas of the brain: frontal, central, occipital, left hemisphere, frontal, central, occipital, right hemisphere. They're represented by the different cortical rhythms and how they interact with chin activation, how these rhythms synchronize with the activity embedded in the chin. And this radar chart shows diagonals, that is the sum of the vectors representing the strength of all the links. So if we have a large diagonal of the radar chart, this means strong interactions with a particular area of the brain. And now we see that again the same stratification. Cortico-muscular interaction strong during wake, weak for REM sleep, stronger for light sleep, and weaker for deep sleep. And this is a very schematic network, but behind this network is very complex, because all these links which are presented here have certain particular profiles. So these are the links showing the interaction of the different cortical rhythms, delta, theta, alpha, beta, gamma, from the central area of the brain with the chin muscle activity. And what is the synchrony between bursting activity in the chin and bursting in this cortical rhythm dynamics? And one can find that there is a very unique pattern, a profile of link strength, so that these links for gamma 1, beta, and gamma 2 are the main mediators in the interaction of this brain chin network. And these profiles change in intensity and change in characteristics with transitions across from wake, REM, light, and deep sleep, and of course very parallel with the left and right brain hemisphere. One can extend this to the brain-eye movement and find different informative dynamic network maps, also following the same pattern: strong interaction, big diagonal, especially the interaction of the eye with the frontal areas of the brain, because you see here that the vectors are stronger to the frontal areas of the brain and the vectors do not reach to the periphery at the occipital area. This is very interesting for vision science. And so again, these interactions are stronger for wake and light sleep and weaker for deep and REM sleep. So this law is quite fundamental, but with very different profiles of interactions. Before we had gamma rhythms to be the main mediators in these dynamic networks, and now delta rhythms turn out to be the main mediators in the way how the brain controls eye dynamics. So we can have different dynamical maps representing different peripheral organ systems: chin, eye movement, heart, respiration, in the same physiological state. And now this is an orchestrated, coordinated behavior among peripheral systems: heart, legs, eye, respiration, with various centers of the brain represented by various cortical rhythms. And again, this large network also undergoes the same stratification, with higher connectivity and strong interactions for wake and light sleep, weaker for REM and deep sleep. So, one, it's not everything connected with the brain. I can answer questions after that. It's not only top-down control. If you take the peripheral systems without the brain, heart, leg activity, eye movement, chin and respiration, they form a small network, but this network has a very characteristic topology. You can see different links have different strength by representing different colors of the links. And so this network reconfigures its structure from point of view which links are stronger and weaker, depending on the physiological state, also with the same stratification between wake, light, deep, and REM sleep. So it's a small movie here showing in real time from a real subject what is happening in these three subnetworks of the global organism network. We have the brain-brain network, the organ-organ peripheral network, and the brain-organ networks together. So here we'll see what's happening with their topology, reorganization, and here these bars show the average link strength of the links that you see in the topology. So here there is a trace in real time about 6 hours, 240 minutes of one subject who is in the beginning is awake, then goes to REM sleep, to light sleep and deep sleep. And so let's see what is happening. And in real time, you see a very synchronous change, both in the connectivity across all these networks and in the strength of the interaction, in the strength of synchrony among systems represented by the bars below. So it's really like a symphony, like an orchestra, how the organism works. And now we are discovering for the first time how these systems really coordinate and orchestrate together to produce a state. And now we know what type of connectivity and what type of link strength and which modules are involved in order to generate deep sleep or light sleep or REM or wake. And so, of course, this can be extended to other studies looking at how this behavior changes with age. So this is the brain-brain network for wake, REM, light, and deep sleep that I showed before, and how this network changes with different age groups horizontally. And we see these same stratification patterns that I showed before in the connectivity and the average link strength. It's preserved with aging, but declines with quite significant percent. So now we know that aging is not an idiosyncratic breakdown in these brain functional networks. Certain modules in these networks are very preserved. For example, this module is well expressed, as well it's expressed in young subjects. This module, frontal to central, is also well expressed and preserved as in young subjects, but other modules decline. So nevertheless, there is a certain basic mechanism of regulation that maintains the organization of the network both at high age of 80-year-old, similar to the young age from point of view of how the network reorganizes with transition from one state, deep sleep, to light sleep, and from light sleep to REM and wake. So this can be dissected for different subnetworks, again, left and right hemisphere with the same universal patterns. And one can also further extend brain-organ interactions. For example, this is the brain-heart interaction represented by these same radar charts for different age groups. So this is the young group that I showed before. For wake, REM, light, and deep sleep, stronger interactions for wake, weaker for REM, bigger diagonal for light sleep, smaller diagonal for deep sleep, and how this gradually declines with aging when you go from young to old, and these are healthy old subjects. So now we can develop hundreds of maps. As a matter of fact, we found out that we need millions of maps to represent these interactions. This is network maps representing brain-eye movement interactions for different states and different age groups. Again, the same age groups here going down, and the interactions decline. And brain-lung respiratory interactions, brain-muscle activity, and also the interactions between peripheral systems without the brain also follow particular same stratification pattern with a particular profiles. So we have here a network of 10 links. Some links are stronger, other weaker. There is a profile, and this profile gradually declines with age, but is preserved. And so there is certain a lot to understand how these networks reconfigure temporally and dynamically in order to generate behaviors across states and age groups. So now I showed before a single radar chart representing here, for example, the brain-leg muscle interaction. And it looks quite simple, but behind this simple radar chart, there is this complex network where the leg has multiple components because the leg muscle has hundreds of muscle bundles with thousands of muscle fibers that have different rhythms of activity, and how these individual muscle fibers and bundles interact with the brain. This is not known to science, but we start to obtain certain first complex networks, and you see how these networks also change topology and dynamics with transitions from basic states to another. Also characterized by particular profiles. This is for the brain chin muscle across areas of the brain, frontal, central, occipital, during wake, left hemisphere, right hemisphere, and we see how these patterns of interaction change depending on the physiological state. So now we start to understand gradually how physiological states start to emerge out of this complex network of networks that involves multiple systems. And of course, it has implications to disease. What I showed before was for a group of healthy, young group of 95 subjects. Now we have a larger cohorts. And we have here a group of about 40 subjects with Parkinson's disease. And you see certain modules of this brain network are very overexpressed, over-synchronized, because you see these red colors of the links. And correspondingly, the number of the links and the average link strength increases significantly. With Parkinson, we start to learn something about brain rhythm interactions. But for example, something that is still known to Parkinson research is how brain-heart interactions change with Parkinson. And we now see this stratification that we see in brain-heart interactions with sleep stages. In the young group, it changes with Parkinson. So this brain-heart interactions, which is quite strong, synchronization and in healthy group declines with Parkinson. It's first evidence. This is still unpublished work. And also brain-muscle interaction also declines in Parkinson. This is somewhat to be expected since we know Parkinson subjects have locomotor degeneration. And these networks, these are the bar charts representing the dynamics and the links embedded in these networks. So each bar represents a particular link. So you see that the profiles of these links changes depending with Parkinson. And in some physiological states like wake and light sleep, they change more than in others. So this all new knowledge that we are acquiring. The same thing for organ-organ interactions. We have profile of organ-organ interaction for different physiological states in the healthy group. And this profile declines in strength, the bars decline, but also the profile changes with Parkinson. Very important. Now we know which pairs of organ systems interactions are mainly affected by Parkinson. So these ideas, as I said, started more or less about 15 years ago. In a work in 2012, we, for the first time in the literature, network physiology as a term was published. A Harvard Catalyst program in 2012 organized for a Harvard faculty in medicine the first symposium on network physiology. This was followed by science news announcements and focus research work in physiological measurement in the Journal of Physiology and so on. Physics World in 2016 devoted a cover page on the field of network physiology saying body functions, the new science of network physiology. And they asked a very important question: can we map all information being circulated in the human body, and would doing so be of any use? A very good question. Is there any use to all this done? But now we start to understand that in addition to knowing the static atlas of the human anatomy that took about 400 years from Leonardo da Vinci time for modern science to develop, and it's hundreds of maps, thousands of pages. So we want to develop, and the vision is to develop a dynamic atlas in addition to the static ones, showing the interactions of these systems with hundreds and preliminary calculation. This would be several million maps that would transcend dozens and hundreds of healthy conditions, age groups, hundreds of physiological states, and thousands of clinical conditions. So can we understand better heart failure? Can we understand what means coma? Because in coma, certain functions continue unimpaired and other functions are impaired. So what means heart, multiple organ failure or coma or other type of diseases? And not only by representing beautiful maps, but also finding the critical zones and beyond which these interactions start to fail and the whole network break down. And data is essential. We know that before the human genome, there was certain sporadic data finding metabolic changes in the human DNA and certain associations here and there with certain diseases. But after the human genome was scored and sequenced, there was an explosion, proliferation of knowledge and science. So now the human genome is a gigantic database of 3.5 billion data pairs, base pairs. This is roughly a library of 1,000 volumes, 1,000 books with 1,000 pages covered by letters. of the different nucleotides, 3.5 billion data points. Now, the human body, if generates every minute, every second, multiple data that we still not harnessed today in modern science, both in basic science and in medicine. The brain, various physiological parameters of the brain, various physiological blood pressure, oxygen concentration, body temperature, various muscle activation, hormonal functions, kidney function, liver function characteristics. If you take just 100 parameters in one 24-hour day, and you record them continuously, synchronously, with about 100 hertz, which is nothing in contemporary physiology and medicine, you'll get one magnitude more data points in 24 hours, one magnitude more than the human genome. In 1 year, 10 to the 12. In one lifetime, almost astronomical numbers. So the way how it took about 50 years from the discovery of DNA to make association with mutations, genomic mutations with disease, to trigger the necessity to sequence the human genome, and now to have networks of different diseases based on overlapping genomic content, and we are going in the future to big super data of personalized genetics, we envision that network physiology, we are roughly right now building initial steps in first maps, in the future can lead to developing the human physiolome project, this atlas of organ interactions across states, cross conditions from the organism level to the subcellular level, integrating also effects of drugs, clinical treatment, monitoring devices, and also personalized medicine. Implications for new devices, of course. In intensive care unit today, in the most advanced hospitals, we have separate devices. These devices do not integrate data. This is something that has to be done in the future, and we are working in this direction. In pharmaceutics and pharmacologies, we develop drugs that are targeted for a particular tissue, particular cell, particular organ, but very often they misfire. And the question is, can we assess their effect of interaction among systems? We need, of course, all this information can revolutionize personalized health monitoring. And of course, we need new education, both in basic sciences, physicists who go in this field, make new discoveries, as well as physiologists and clinicians who are trained to read and to understand and to utilize this information in clinical medicine. And of course, implications for new AI and robotics, where all these principles of network physiology could be integrated in robotic devices or swarms of bots, or even the new cyborg type of elements. So this is really a multidisciplinary work. Here is a map of Boston, our lab, mainly doing computational basic research. We collaborate with colleagues in Harvard on multi-sensor type of networks with which we need to collect simultaneous data. Physiologists at Brigham, at Harvard Medical School, clinicians at intensive care at Mass General, and so on. And of course, it's an effort of many people. So I understand time is over. I just want to invite you to regular meetings. We organize every 2 years. This year is in July, end of last week of July, 27th to 1st of August, and a very beautiful place in Lake Como in North Italy, next to Swiss Alps, where we will discuss all these aspects of network physiology. A new journal has emerged 4 years ago, now with a quite substantial editorial board and 11 sections covering various sub-areas of research in network physiology. I invite you to just check it on Google Scholar, and thank you very much for your attention.
Thank you. This was really a fantastic talk. So I request the audience to type your questions in the Q&A box at the bottom of the Zoom. And
if you don't mind, if you have some questions, would you read them for me?
yeah,
if it's
already
okay.
there are some questions. The first question is from Joseph Thomas. Can network physiology be considered a systems approach to understanding human organ function?
Can network physiology help system approach?
Yeah, can it be considered a systems approach to understanding human organ function?
Right, yes, it can be understood as a systems approach. We need to understand, not up to now the focus is we understand physiological function or physiological state or global behavior of the organism from point of view of single organ. You look at the brain, And you say, okay, in deep sleep, the brain has dominant delta rhythm, or the heart has low heart rate, or the oxygen concentration is this and that, or the muscle tone is low. Now we start to say, okay, this is useful information, but not sufficient to fully understand how these systems integrate. And I mean, Robert Thomas is one of the most eminent experts in the sleep medicine, clinician. We are, lucky to have such a person being interested in network physiology. So indeed, I believe now we start to see also preliminary studies, which I didn't show. If we have sleep disorders, we see how these interactions as a network start to change with sleep disorders. So it's very important I think it will have impact on sleep medicine. But system integration is the key, and knowing how systems integrate dynamically. Again, these are nodes and networks which are different philosophy here we have than the complex networks that have been studied the last 30 years. I want the audience, I will take 1 minute to explain this. What is the difference between the networks we have here and why this is not just an application of complex network theory to physiology? Because in complex network theory, we consider the nodes static, they're just a dot. And we consider the links also static, they're just a link. Now, we answer the question there, association of network topology with network function. But the function is a fixed topology with fixed links. Of course, the links can have a distribution of strength, but once the choice is made, which links have what strength, once distribution is chosen, you have a static topology. Now you say how information flows on this topology. And from point of view of dynamical aspects, complex network asks the question, what if I remove several links? Will the network disintegrate? What if I add several nodes? Will the network improve in its function? So this is the dynamics. But what we face in physiology is a different problem. I may have this network which has fixed topology, same links, and same connections. The topology doesn't change. And now I change the dynamics of the nodes, and I change the dynamics of the coupling between these nodes. The topology doesn't change, and yet I may have a new type of state emerging in such a network. And so how to define global state on a network of dynamical system with elastic dynamical links, this requires a new theoretical framework. And as every discipline, which is a real discipline, each discipline needs a new theoretical framework. So this also in the context of the question from Robert Thomas.
Yeah, the next question is from Andrea. Thank you for the interesting talk. How does network connectivity between brain and the peripheral system change with different sensory inputs in the awake individual? Or does the connectivity change with peripheral sensory stimulation?
Yes, so now there are certain pioneering studies now in this direction. And if I would paraphrase the question, if I have a cognitive task, and in cognitive science, neuroscientists focus on the brain, certain neuronal populations involved in that. And so now I sit in a chair resting, and then I sit in the same chair in the same environment, and I do a cognitive task. So there is certain input. So, and we find now that not only functional brain networks reconfigure, but also peripheral systems, brain peripheral and peripheral systems also reconfigure their dynamics. So there is a lot to study in this direction. This area is practically untapped, but there is some indication that even for cognitive function, we involve not only sub-areas of the brain, but the whole body integrates in order to stimulate this work of the brain, whether through your respiration, heart activity, muscle tension, and other things. This has also, of course, now more studies go in exercise physiology, how we can integrate one large group of muscles. Human body has about 900 muscles, different type of muscles. Each is a separate organ. And we perform various random movements or repetitive movements throughout the day, and how these movements emerge of coordinated synchronization among muscles. and not only among muscles, but among the muscle fibers, thousands of muscle fibers inside each muscle. And so how does this impact fatigue? Can we train better elite sportsmen? Can we use this in military? So there is a lot of questions now we start to answer, and network physiology has gone also in an area called network physiology of exercise. So there is a lot to do. It's a field in its infancy, and if many of those listeners today, I would invite to join us and to contact us and join our meetings in Como or in other forums.
Okay. The next question is also from Andrea. It is, how can we use the fractal nature of physiological signals to definitively conclude a certain disorder, given that there could be several confounding mechanisms? Thinking about translational approaches using these measures to diagnose disorders, what's the way forward?
Right. So fractal characteristics of dynamics of individual systems. We have, this is a very important question. The last 30 years, there are already a large volume of studies in the literature on that. Now we can say that practically every physiological system, whether it's an organ system, whether it's at the tissue level or cellular level, exhibits certain fractal dynamics. And this means that multiple scales are involved in order for this system to function. This means that multiple feedbacks regulate the mechanisms that control these systems or subsystems, whether it's the cell, the tissue, or the organ, or the whole body. So now, that said, this change in fractal dynamics, this slide in the very beginning of the talk that I showed, exemplified exactly this. It showed a scale invariant bifurcation of variability at different scales, which is fractal characteristic of cardiac dynamics under healthy condition, and then it breaks down with certain pathology, becomes repetitive. There is no repetitive. This means only one or few scales are involved. So the fractal involvement of multiple scales in the dynamics breaks down. So when a system is not involving multiple scales, then the system is sick. Now, this is about individual dynamics of individual systems. Complex, of course, fractal, multifractal, multiple scales. But now, how these multiple scales in the dynamics of one system cross-communicate with multiple scales in the dynamic of another system, especially when these systems work at different time scales, the brain and the neurons at milliseconds, and the heart at seconds, respiration at longer scales, kidneys at hours, this is still for science to find out. A lot of work in this direction theoretically, but I'm sure it's important because now we know this example on cardiorespiratory coupling. We are aware that systems now can cross-communicate simultaneously by multiple forms of interaction. And so these multiple forms of interaction work at the same time, but they work on different time scales. So maybe this is also a way to facilitate transfer of information from one fractal property of the one system to a fractal property of another system, whether it's heart and respiration, whether it's brain and heart and respiration, and muscle and so on. But still to be studied how the underlying fractal type mechanisms of control of individual systems play a role in the dynamic interaction among systems. Again, these are elastic, adaptive networks. The interactions always change in time, they're transient. And that's why it's very unique, specifically for this audience, which studies phase transition, critical transitions in systems. So the human body is the quintessential example of critical changes in the dynamics and the coupling, and now we see also in the network interactions among systems depending on physiological state.
Thank you. And the next question is, can we derive early warning signals based on body-body, body-organ, organ-organ network dynamics for transition from one sleep state to another?
We hope. So would be nice. There are studies we are particularly in our lab focusing, for example, on people with seizure subjects who have, so that we study not only the brain, but also interaction of the brain with other peripheral systems, especially systems under autonomic regulation, to predict sudden death, SUDEP. I'm sure that there are many other contexts in which this could be, network physiology could be utilized. And I'm aware that people now even use it for cancer and for other conditions, but this most probably will be the future. The way how the logic of science went when we discovered complexity and variability of individual systems, and the immediate question was, okay, out of this variability, can I derive a pattern that can tell me a few minutes before a cardiac arrest or before a heart attack or a few hours before that? Can I predict it? Now the question is, can I enhance these prediction measures by network-based measures derived from network physiology? And I am sure that the more we know, the more detailed measures of prediction will come up.
Okay, thank you.
I'm sure both conceptually and also based on certain preliminary findings that are not published work.
Great. The next question is on comparing coma and brain dead.
Coma and brain dead. Believe it or not, we are working. I personally have interest for almost 10 years now, and there is a lot of red tape to overcome in intensive care unit. People with cardiac arrest are very often or brain trauma, they're very often in coma or in induced coma. And so what is coma from point of view of network physiology, we still don't know. And turns out to be a very expensive question. So whoever asks this question, I welcome them to join. But just to say this would be dozens and hundreds of thousands for just a small set of subjects in the intensive care unit with a lot of red tape to overcome and institutional review board permissions and so on. We are working in this direction. It would be interesting to understand how multiple system diseases like cardiac arrest, for example, is a multi-system disease, sepsis and others. Cardiac arrest is one of the most prevalent cases of subjects being admitted in intensive care unit because it involves the brain, the respiration, the heart, muscle, everything, metabolics. And so can we hook now multiple devices that are produced by different companies? These devices have different softwares. Can we integrate to make them a single platform recording in synchronous way so that we can determine causality between systems of a subject in cardiac arrest and in the same time in coma, or somebody with brain trauma and in coma? And I think this would be really, really a big step forward in clinical medicine. But still to do, on the list to do. And to get substantial funding for that, because these are questions that involve really serious funding.
Okay, with that, the questions in the question Q&A box are over. Does any of the, oh, there's one more question. Yeah, there's one more question. How effective is phase synchronization analysis compared to other traditional methods such as coherence analysis when analyzing data like EEG?
Phase synchronization analysis is very effective for systems that have underlying well-defined oscillations. So now for systems which have a strong stochastic component, then it would not be very effective. So now again, for systems which have embedded oscillatory component, and that these components are not very much mismatched. So I am aware that it was a very, very big discovery what Pikowski and Rosenblum and Jürgen Kurz have done 30 years ago, because they taught us that two systems which are nonlinear in nature and in chaotic regime individually can be coupled, and now I can have their output signal, which usually traditional methods cannot tell me whether they're coupled or not. But with the synchronization method, even in the condition of nonlinear chaotic regime, I can deduce based on the output signals, that these two signals come from systems that are coupled. This was a tremendous step forward in nonlinear dynamics, and this had multiple implications for lasers, for everything. For climate, for multiple physical systems, and now we see in physiology. But one has to be at the same time aware that there are limitations, because first, physiological systems do not interact with fixed coupling, and second, the method in general is sensitive to how narrow are the bandpass filtering of the data that you do to pre-process physiological signals before you do your analysis. So there are things one has to be careful, but when you apply it to systems of similar character, so phase synchronization is very, very useful. Now, when we have systems with very distinct dynamics, and that's what we observe when we go from one peripheral system to another, then it's a very big question. Can you have a method that can work on multiple scales and work with multiple dynamics at the same time, to be sensitive enough to discern transitions in these couplings and transitions in these interactions? So, and by studying various methods available in numerical recipes on the shelf, in applied mathematics, computational methods, we had to devise this time delay stability method, because others would show a lot of limitations. And these two slides I showed in the very beginning show this. Actually, I can maybe show this back to the person who asked this question. Here, okay, where was it? Okay, here. So here you have two signals. They have their underlying dynamics, and they have superposed bursting activity. So now, any method you take, if I give you two random signals, but with synchronized bursts, every method, transfer entropy, coherence, even synchronization, will give you nothing. They will not identify the presence of synchronized bursts, because the two signals in nature are white noise. But still they have synchronized bursts. How do I dissect the presence of synchronized bursts? And in that sense, time delay stability turned out to be a useful approach.
Okay, so
just to say this, not to say that there are many applications that are relevant for all these methods, but people develop Granger causality, new approaches, particularly for Granger causality, transfer entropy, and so on. But for signals with embedded stochastic element and synchronized bursts, this is what we see both in brain dynamics, cardiac, muscle activity, or whatever. To identify the presence of synchronized bursts in the context of a certain noisy signal, especially if the signals are very weakly autocorrelated, then this is a very big problem. And also, not many people in the literature know a very fundamental fact, that if you take two signals coming from two systems that are not coupled, but the signals are autocorrelated individually, if you simply cross-correlate them, you'll find very strong cross-correlation. But this means nothing. It's strong cross-correlation just because the signals are autocorrelated. For those of you who are experts in this field, most probably they know. Those who are new in the research, do not cross-correlate autocorrelated signals, because you'll get strong cross-correlations. And this would not mean necessarily that the systems are coupled. In that sense, the synchronization method developed by Kurtz and others has strong advantages.
Okay, there's one more question. What do we know so far about body periphery interactions in behavioral disorders such as attention changes, depression, etc.?
Well, depression is very strongly related with physiological functions, both at metabolic scale, both at sleep, also with physical activity. So, this I would just say that this is a new area to be studied.
Okay, thank you. I have a question. using the networks, any kind of control is possible so that somebody who doesn't sleep well can be with some electrical signals can be made to sleep well, and using these networks.
No, now we know. Now we know better. Let me redefine this. I don't know what means well or not well. Some time ago, people thought, so let's say 15 years ago, people thought that arousals during sleep are always something negative. It interrupts sleeps, fragments sleep, and arousals are bad. Now, in the last 20 years, we discovered that arousals are essential part of sleep regulation. We need arousals to have sleep. So now, when you ask me what is a good sleep, 20 years ago, I would say, a clinician would say sleep that doesn't have arousals. Now, people will say, okay, no, good sleep is a sleep that has arousals that are organized in a particular criticality way. And when these arousals are perturbations from the environment or do not follow the laws that are natural for a good sleep, then you have low quality sleep. It's the same question with network physiology. Now we learn what means deep sleep or what means light sleep or wake from point of view of interaction between systems. And these interactions can be expanded. I showed just a couple of systems here. What about metabolic interactions? What about other aspects? What about muscle interactions, or many other questions could be entered here in the framework of the network. But now we know that by knowing the structure of the network, by knowing the hierarchical organization of the network, we know that, okay, we have one hierarchical network organization for deep sleep and a different for wake. So now I can define what means good deep sleep and what means good light sleep. And now I can also define what means insomnia or what means narcolepsy. And there are research studies going exactly in this direction.
Great. Thank you. I think we are done with all the questions. So once again, let me thank the speaker for this very wonderful talk. Thank you so much.
Thank you.
And we will take a break for the next 3 months because it's summer. So the next webinar will be in September. I will send the announcement to everyone. So thank you all and have a nice day wherever you are. Bye-bye.
Thank you, Professor Sujit and Professor Kurz. Thank you again for your invitation. And for all the audience for their time.
Thank you.